Hello, everybody. I hope you are in good health. I'm going to present work on automation and robotics for high-throughput phenotyping and precision horticulture. First, let me give you a quick introduction who I am. So my name is Rick van der Zedde. I've been working for 15 years now at Wageningen University in research in all kinds of roles, actually. And recently I've become the vice chair of the International Plant Phenotyping Network. So I have a background in AI, and I hope today to inspire you by sharing some applied research projects and new ideas. So first, let's introduce actually the topic. So we are focused on the phenotype. But if we want to measure how a plant grows, we really need to understand what is actually its genotype on the one hand, but also we need to very carefully figure out how the plant is actually situated, so in which kind of environment. And we have presented this also to the Dutch Science Organization and said we need a serious amount of money to actually study this equation. And then they decided actually to fund our new phenotyping center called NPEC, which I will show the outline of also within this presentation. So I think the challenging part of phenotyping is that it's a very multidisciplinary domain. So we need people from biology to really understand the plant itself. We need people from high tech to be able to deal with, for instance, AI tools, computer vision and robotics. But on the other hand, all these tools generate a massive amount of data. So we need to have data scientists also on board. If we combine these teams and these people really understand each other, then we can say we are doing decent phenotyping research. But there is another challenge actually that I want to bring up. If you look at the technology adaptation cycle, I'm afraid that actually the people from technology also present at this tiki crop are the innovators. Potentially the data science people are the early adopters, but if we want to get real impact with all these novel tools also shared at this tiki crop conference, the majority of the biologists are still a bit hesitant. So they might be the early majority, or maybe even the late majority, but then the magic actually happens. So quite a few of our projects are still actually in this gap. So they're not embraced by a large part of our community. That's really needed. That's also where we are aiming for the NPEC project. Because NPEC is a large-scale research infrastructure project. So we are building a research center. We will open up and we offer access to anybody interested. So we can join up, team up on all kinds of topics. For instance, automated screening tools with novel sensors or AI tools, or we can implement big data approaches. Everything needed to get a good understanding of the plant and actually boost our plant science domain. So the essence of NPEC is that we have several modules in which the phenotype is studied. In ecotrons, we focus on plant microbe interaction. We want to have a range of environments offered to the plants and figure out how and why they grow differently. We have high throughput phenotyping in climate chambers and the greenhouse in which we can study plants, but also of course on the field. I'm going to zoom into these three modules today. So module number 4, the climate room module, there we are talking about a large number of plants. They are constantly monitored with a range of camera tools. This is actually an existing climate room that we have at our institute that has been running for actually more than a decade. And it's really time to do a decent improvement on the technologies used in this room. Because of timing, I need to be quite brief today, unfortunately. So this is actually the outline of the module number 4. So 5 climate rooms with all kinds of phenotyping tools. And if you want to know more, you can also go to our website. Current status, the building is actually already there, but the equipment will come next year. Talking about the NPEC greenhouse module. Here I'm going to zoom in onto a more specific example. So we are aiming for a greenhouse in which we can study the plants in multiple environments. So each compartment has its own settings with a range of cameras and tools. Currently, the greenhouse is already there. It's actually waiting for the equipment to come. So this is the internal of the climate room of the greenhouse, to give you a few impressions. And I want to zoom in onto one specific camera module in there, a module in which I was also myself very involved. We want to capture the shape of the plant. So we're using a multi-camera setup, in this case 15 cameras, that take pictures of these plants from all angles around the plant in different layers, in this case 3 layers. And we also would like to add additional information, for instance with the near-infrared camera, to that plant. It's conveyor based, so plants go through the imaging cabinet, and then we can rapidly, like within a second, generate a 3D model of, for instance, this potato plant to measure all kinds of aspects. And this particular 3D technology has also been used inside a robot that has been embraced by industry, by breeding companies, to actually speed up the sampling of young seedlings, where a piece of leaf is actually grabbed by the robot, and the robot is steered towards the right leaf by this 3D algorithm, which generated this 3D model on the left. So the last topic I would like to mention today is our field developments. So we have been working on field robotics for years now, also in combination with drones. First, I would like to show you a prototype developed a few years ago by my colleague Gerrit Polder and quite a team. This cabinet is mounted on a tractor. It contains a range of cameras, hyperspectral camera, 3D camera, RGB camera, and all data is accurately registered on GPS location. And then the aim, for instance, in this particular project, was to scout for disease symptoms in potato plants. So this is a picture of one of the runs that we made. This is a top view line scan picture from one channel of the hyperspectral camera. And the essence is actually that we have an input image, a range of channels from the hyperspectral camera, top view. We are using AI, deep learning, to actually translate this picture into simply a yes or no prediction. Is there a disease plant or not? And we have been collecting data in which experts were actually indicating the position of the disease plants. So we have a prediction and a target. And this picture is more clear. So the top picture shows the exact locations where the expert said, yes, there is a disease plant. And then we trained the AI, and the second row is actually output generated by the AI, where we can see that the AI is trying to mimic the expert. So I'm not able to go into details today, but all the work has been published in this article, and also the actual data is now available through this link. And it has also been written down in the Vision Systems last year. But there is one striking thing here. This device was used by this particular research team. But what if you would like to use this device? Then it's not so easy actually to get access to this quite sophisticated toolbox. Therefore, we also decided to move forward actually and make this particular approach available to anybody interested through actually this device, which is part of NPEC. So this is the FieldExplorer. And we produced a short video to show actually the essence of its functionality. So it's actually developed by a company, specifically on our requirements. So it is operated by you could say, a service engineer, an engineer that is assisting the researcher with getting decent data. Also on this vehicle, there's a range of sensors, and all the data is captured on plot-specific locations. And actually the driving is easy. So the steering and also the speed is regulated by the device. And our aim is also in the upcoming years to combine the data from the drones with the FieldExplorer, which offers up a huge range of opportunities to fully grasp the phenotype of the plant. So summing up this actually short presentation is that we really are focused on measuring, understanding, predicting plant quality developments in all the ranges of, you could say, environmental control, climate rooms, greenhouses, fields. And we are focused on setting up new research projects to explore these new tools. We are also offering access to anybody interested to use these tools for their own benefit. So we are exploring the potential of large-scale research infrastructure for plant phenotyping. And we really needed to be aware of our different backgrounds. So let's explore collaboration. So hopefully you have a few questions for me. This was a pre-recorded version, but I will be live available to answer your questions. Thank you.